So, can you hear me? Yeah, okay. First, I would like to start by thanking the organizer of this amazing meeting for giving this opportunity to talk about rabbit domestication. Well, I put the original title in a parenthesis, but I decided to use this one. The genetic basis for tameness revealed out of a rabbit hole. I'm sure that you can recognize the picture from Alice in Wonderland, who chased the rabbit. We also chased it, but not to get to Wonderland, to find some information, some explanation about the questions that we had. Okay, within the last 25 years, there have been brilliant studies trying to identify genes and mutations that have altered the phenotype of domestic animals. But the genetic changes associated with the initial steps of animal domestication are still poorly understood. What is certain is that the behavior should have been targeted by selection, allowing the animals to survive and reproduce under conditions that would be too stressful for wild animals. But why rabbit? Not because it's a cute animal, but from agricultural perspective, it's very important for meat production and wool production. It also has been extensively used in biomedical researches and biotechnology. And also, it provides a unique opportunity to study the genetic of domestication because it's a relatively recent event and we know where it has happened. I come to that in next slides. The domestication sites is still densely populated by wild rabbit, which is expected to resemble the ancestral population. And the wild rabbit had a very restricted geographic distribution. It was interesting for Darwin. This was an interesting species. As he says, hardly any animal is more difficult to tame than the young of the wild rabbit. Scarcely any animal is tamer than the young of the tame rabbit. And here you see the wild rabbit. You can't really get close to him. They just escape. But here you see the domestic rabbit, which is accustomed to human presence. This species abundant in Iberian Peninsula, and the climatic changes led to allopathic speciation of two subspecies. Algirus in southwest and northwest we have Cuniculus, which later colonized southern France. And how did it happen? It has an interesting story. Monasteries in southern France started keeping rabbits after the Pope declared young rabbit, not as rabbit, but as fish. So, yeah, so it was possible to eat during Lent. I don't know how he came to this conclusion, but anyhow, by 17th century, thanks to him, by 17th century, phenotypic diversity in colors and sizes were present. And in this painting from 16th century, you can see a domestic rabbit. And objectives of this study were first genome assembly and second whole genome resequencing to identify loci involved in domestication. Here we have 14 wild populations, 11 from Iberian Peninsula and 3 from France. We had 6 domestic breeds together with reference individual and an outgroup with no short hair. No previous study on animal domestication has involved such an extensive characterization of genetic diversity in the wild ancestor. The assembly was part of a 29 mammals project where a female from Thorberg, New Zealand white line was sequenced, and the assembly size is around 2.7 gigabytes and N50 size of 36 MB. And the sequencing was done using genome analyzer 2 machine at Broad Institute, where we received paired end reads with 76 base per length and 10x coverage per pool. SNP culling was done using VariScan and MPyLab, and after stringent filtering, we got 51 million SNPs. So it's very polymorphic genome. And you can also see that in nucleotide diversity, especially in wild Iberian, which is close to 0.1, 0.01, sorry. And after the first bottleneck, where the southern France was colonized, you can see a drop in nucleotide diversity, followed up with another bottleneck after a domestication event. But still, you see nucleotide diversity in domestic populations. We compared the populations to reference genome by identity score in 50 kb window. And here you can see the reference individual, so it's identical. And all the domestic population show high similarity to a reference individual. And here we have wild French populations, which are slightly different compared to domestic animals, domestic groups. And if you walk, well, you can't walk, it's long distance. If you move from southeast to northwest, you can see a gradual decrease in identity score. So by looking in this figure, you can see that it's indicative that wild French population can be ancestral population of domestic rabbit. And here we also compared the reference allele frequency between domestic groups and wild French groups. Here you can see strong correlation between allele frequency, where most of the SNPs are carrying the same allele in both populations, and there are few SNPs went to fixation in either of groups. We had many differentiated SNPs at non-coding regions, and how we calculated that. First, we measured the delta allele frequency between populations, domestics and wilds. And here you see the distribution of number of SNPs in each delta allele frequency bins. And there were only 20 SNPs out of 51 million SNPs went to fixation. But then we asked, where are these high delta allele frequency located in relation to features of genome? So here you see the same delta allele frequency bin, and here we calculated the M value. And here you see that there is no overrepresentation in intronic region, but you see overrepresentation in coding and UTRs, and especially overrepresentation of high delta allele frequency in conserved non-coding elements. So the next question is that, what about the exonic SNPs? Are they affecting any gene expression, any gene inactivation? So there was not a single example of gene inactivation mutation with high delta allele frequency. There were only 14 missense mutations with high delta allele frequency above 0.9. But just a few candidate causal mutations were detected. For example, TTC21B, where glutamine has been replaced by arginine, and all the placental mammal show the wild allele, and none of them show domestic allele. And the same for KDM6B. Majority of placental mammal show the wild rabbit allele, but not domestic, where arginine has been replaced by tryptophan. And these two genes are very important genes in developmental stages. So we then ask which genes are associated with extreme SNPs. We had 1640 SNPs at conserved non-coding sites with a high delta allele frequency above 0.8. In order not to inflate the signals, we chose one SNP per 50 kb. And with this subset, we performed enrichment analysis using great software. Here you see three types of information: number of genes in each category, enrichment value, and Bonferroni corrected P value. And so we have high number of genes in each category. And you can see higher enrichment. And the top hit was central nervous system, brain, with P value 10 to minus 25, so it was very significant. But what happens if you knock them out in a mouse? Using MGA data, you can see the same type of information. And here we have a maximum 250 genes in each category, in categories that you can see here. And the top hit was abnormal nervous system morphology with p-value 10 to the minus 18. It's significant. And once again, we see genes which are expressed in central nervous system. We did the same thing for, we did the same gene enrichment analysis for GO terms, and self-fate determination showed the strongest enrichment. And the extreme SNPs were located close to these genes in this category. BMP4, catenin B1, I absent homolog, and KLF4, PAX2, 61, SOX2. We performed an EMSA analysis, and 7 out of 17 observed probes at SOX2, KLF4, and PAX2 with extreme delta allele frequency located at conserved sites showed differences between genotypes. Here is an example of one of these teams close to SOX2, and you can see domestic rabbit is the only animal showing different nucleotides compared to other species, which you can see here. So it's, you can see a marked gel shift in comparison between wild allele and domestic allele. So concluding the studies that we have done is that our results is consistent with a highly polygenic background for rabbit domestication. And there were very few fixed differences, rather shifts in allele frequency representing soft sweeps. It shows that the selection has primarily acted on a standard genetic variation. There was not a single example of gene inactivation in our data. And as you know, changes in non-coding sequences are numerously much more important than changes in coding sequences. In our representation analysis that we done, we could see that genes involved in neuronal and brain development which is consistent with selection for tameness. Then the question is that if there is any domestication gene. Well, we propose that they may not exist because tameness has highly polygenic background and evolved by shifts in allele frequencies at many loci rather than by critical changes at only a few domestication loci. Last but not least, it implicates that feral population may adapt to the wild by back selection at loci that have not gone to fixation. And here I would like to finish my talk by acknowledging people who have been involved in this study from Uppsala University. A special thanks to Professor Leif Andersson who led this project, people at the University of Porto, and the Broad Institutes, and the Rabbit Genome Consortium are founders of this study. Thanks for your attention.
Eric?
So presumably the domestic rabbits are escaping sometimes and maybe breeding
wild
with,
rabbits.
Sorry, but like domestic rabbits might escape and then breed with the wild population at times. Is that a phenomenon that occurs?
Sorry, I think...
actually, I'll just explain the whole question rather than asking pieces. So if these domestication loci are helpful for the domesticated animals, it might be risky for the wild ones. And if so, have you looked at those loci to see if there appears to be resistance to their incursion by escaped domesticated animals that then would breed with the wild animals they're related to?
We have not really tested that. I can't really comment on this question, sorry.
Okay.
So there's strong ongoing selection in cattle for behavior and domesticability, I guess, or ongoing domesticability. And one class of genes that we see very strongly enriched for selection in cattle are associated with human autism spectrum disorders, which is interesting. And I'm wondering if any of the genes that you saw were associated with autism spectrum disorders in humans.
Yes, we had some genes, I can't recall the exact name, but we had some of the genes which were involved in the autism disease.
Very nice talk. My question was, how big is the phenotypic difference in, say, meat production, fat layers, and things like that, the other aspects of potential domestication in domesticated rabbit? And do you see any signature from where energy allocation is going in the rabbit, or is it just these behavioral aspects?
Well, in each domestic breed, let's say. So we have been a selection for diversification for specific traits, but it's like a consequence after the domestication. But and in a domestic breeds which we have selected, they are representing different traits which you mentioned.
Beautiful talk. One question I have is, there's a lot of body size variation in rabbits, right? There are these huge rabbits. Did you guys sequence any of those and identify putative loci that could be involved in body size differentiation?
Well, the aim of this study was not to detect those, but yeah.
But you did the sequencing, right?
Yeah. There are some selective sweep for specific traits for each breed, but I can't really, I don't know their genes, which are, yeah. No, I mean, I don't know that.
Any more questions? Okay, thank you very much.
Thanks.